Today we have the great pleasure to hosting Jonathan Fallowfield, which is a professor of translational liver research, which is very catching, at the University of Edinburgh, Scotland, UK. And Jonathan was kind enough to accept our invitation, and Lucia handled all the paperwork, and we end up with a very catching title. The title is SteatoSITE, a gene to outcome data commons for NAFLD research. So Jonathan, please, the podium is yours.
Okay, thank you very much, Claudio, for the invitation. I'm just going to try and share my screen. Can you see that?
Yes, clearly. Go ahead.
Okay, you got that? Can you see and hear this? Can you see the slides and hear me okay?
Yes, we
okay,
can
clearly.
see.
Go ahead.
Perfect. Yeah, thank you again for inviting me. So I'm a clinical academic hepatologist in Edinburgh, and it'd be great to get some Italian collaborators off the back of this. Don't have enough. Some of them have moved to the UK. So the topic of my talk is SteatoSITE, a gene to outcome data commons for NAFLD research, and I'll obviously explain that in more detail in a minute. But this is really want to tell you a bit about what we've been doing over the last few years here. It's been a journey, as you might say. It's been a difficult process, but hopefully what we have got at the end of it is useful for the whole research community that's interested in non-alcoholic fatty liver disease. So I've got a few disclosures which aren't really of any relevance to what I'm talking about today, but I do want to say two things and a slight apology. So I can't talk about everything I wanted to talk about today because I'm embargoed a little bit because of EASL. So we have some abstracts in EASL, and I'm not allowed to present anything that's in those posters and talks. So I've had to take a few things out that I did want to talk about, but I can talk a lot more freely if anyone's at EASL. And the other thing is we've also got a paper under review at the moment. So I'm, again, being a little bit cautious about some of the things, but hopefully there'll still be plenty to talk about. So I'm based here in Edinburgh, and for someone that does translational research like me, I'm in a really good place because all on the same site, we have the main hospital here in Edinburgh, the Royal Infirmary of Edinburgh, and that's where the Scottish liver transplant unit is. So anyone in Scotland that has a liver transplant has it done here. We've also got the Children's Hospital here, and research institutes, the medical school, clinical and preclinical and research imaging. Our clinical research facility is embedded in the hospital for clinical trials and particularly invasive procedures. We have our health data sciences and clinical trials unit up on the hill. We have a bioincubator, so companies can base themselves here in this area of medical and scientific excellence. And then I'm actually about to move to this area here, which is the Institute for Regeneration and Repair, tomorrow morning, in fact. And I've already had my photo taken there. So in a good place to do translational research. And I'm not sure whether everybody knows a lot about non-alcoholic fatty liver disease. So just a little general introduction first. It is, an integral component of the metabolic milieu that we see in people who've got obesity and type 2 diabetes, although there is this lean NAFLD variant where some people who have a non-obese phenotype have a very fatty liver. It's now the main reason for secondary care hepatology referrals. It's most of what I see in my outpatient clinic, although it's different to what I see on the inpatient ward. We're all aware that the prevalence is high already, but also increasing every year. We do have quite a good understanding of the pathophysiology, but we still have gaps in our knowledge, and we do need to learn more about the genetic and the environmental risks. We know that the progression to cirrhosis and clinical outcomes is completely variable and very unpredictable in an individual patient, but in general tends to be a slowly progressive disease course, and it's actually quite unusual for someone with fatty liver to progress to a liver-related outcome or death. So there's a definite need to identify subphenotypes of the disease that have different outcomes and different risk of outcomes. Cardiovascular disease is the main cause of death in these patients. And because of the shared comorbidities, it really is very much a multi-system and multidisciplinary condition. The main unmet need, clinical unmet need in this condition is for biomarkers, particularly prognostic biomarkers that tell us what might happen to people, and also the lack of specific therapies that we can use to treat these people, although you will be aware that in both scenarios there's an intense activity both from companies and also academia. So taken together, it definitely represents a major public health and a precision medicine challenge. So it is a global disease as well. No part of the globe is unaffected by non-alcoholic fatty liver disease, although it's commoner in some areas than other. It's more commoner in South America and the Middle East than it is in Africa, for instance. In Europe, where we are, it's the prevalence rate is around 24% now. This is mainly being driven by the epidemic of obesity and also the explosion in number of people with type 2 diabetes. And because of this, and because of the scale of the disease, in Europe alone, there are huge both healthcare and societal costs related to the disease. And some of you may have seen the paper fairly recently from Jeff Lazarus, which looked at how prepared we are as a planet to cope with NAFLD. And they looked at policies, guidelines, public awareness, epidemiology, research, care and management of patients. And really, the short answer to the whole paper is we're not prepared as a planet to take on this condition, and no country had a proper national or subnational strategy for NAFLD. The prevalence estimations in the coming years are frightening, to be honest, quite staggering. Whether you believe this type of statistical analysis or not, this paper indicates that perhaps by 2040, over half of the world will have non-alcoholic fatty liver disease. So watch this space. The other thing we do know about this condition that's important to mention now is that it is a condition that can progress, but also we know it can improve spontaneously as well, and it does tend to wax and wane. And biopsy series have shown clear cases where there's been progression over time, and also cases where there's clearly been spontaneous regression over time. And so by particularly studying clinical trial data in patients that have received placebo in clinical trials, people have tried to get a handle on the natural history of the disease. And you can begin to separate patients into slow progressors and rapid progressors, and really understanding this and delineating this properly is really the holy grail in terms of biomarkers and risk prediction for this condition, because then we can focus our resources on the people that need it the most. The pathogenesis has been very well described by many people, and this is just a cartoon from an excellent, fairly recent review by Scott Friedman, showing that we do have an understanding of the main pathways, and by understanding the pathogenesis, we can then start to target specific limbs of the pathogenesis, although we don't yet know what the hierarchy of targets are, so where we should necessarily be focusing our efforts. As many of you know who are working in science, there's been an explosion in multi-omics in every disease, really, and NASH is no different. And it is starting to give away some of its vulnerabilities potentially to treatment by both GWAS studies and transcriptomic studies. We have an interest in breathomics, so studying compounds in exhaled breath. Other people have looked at lipidomics and metabolomics and proteomics. So there's a wealth of data we're beginning to unearth, but the key thing that I always consider when I see a new publication is this particular signature or observation a cause, or is it a consequence? And what are the prognostic associations? And that certainly is something we don't yet understand as well as all the data that we're unearthing. For sure, liver fibrosis seems to be the main and the only histological predictor of adverse outcomes. And this is nicely exemplified in this paper from Arun Sanyal, who reported the first prospective observational study in NAFLD. And this is a good number of patients, over 1,500, so 1,773 patients. And they were followed up for a median of about 4 years. And you can see that patients with more severe fibrosis, so advanced fibrosis F3 and cirrhosis F4, these are the patients that have much higher risk of dying or developing liver decompensation events. And this kind of data has been replicated in different cohorts as well, including ours, which I'll show you in a bit. The corollary of that is that if you can improve fibrosis, you may well be able to improve clinical outcomes. And this is data from 2 clinical trials that actually failed to meet their primary endpoint, but nonetheless have enabled us to learn something about the natural history. So in this aggregated cohort of patients, people who had fibrosis regression within the clinical trial had around a 6-fold reduction in risk of liver-related events. So I think this is certainly encouragement that if we can develop effective antifibrotics or reverse fibrosis by other means, then we may well be able to improve outcomes. Those of you that have any interest in drug development will be aware that there is this so-called NASH graveyard now, where we've had many drug candidates and drug programs that have closed in recent years due to lack of efficacy. And you can see many of these here have failed at quite advanced stages as well. I guess that the kind of slight shining light at the moment is the recent data emerging from the Madrigal drug, the THR receptor beta agonist. So I think let's keep all our fingers and toes crossed that this drug makes it all the way. But I think it's been difficult so far to develop effective drugs. So, I mean, you might think we haven't got any good drugs yet. We don't really have any biomarkers that can predict outcomes. We have this massively increasing global population. Is it time to get back to the drawing board? How on earth can we tackle this condition more effectively? And I was struck by this comment that I've highlighted in this review article from Scott Friedman and Massimo Pinzani last year. And they say here in their article that for more refined treatment of NASH, orthogonal approaches that integrate genetic, clinical, and pathological data sets may yield treatments for specific subphenotypes of the disease. And when I read this, I thought, brilliant, because this is what we're trying to do with this project, SteatoSITE. So that was music to my ears, and I think they're right. So we established this consortium, SteatoSITE consortium, and it was funded by Innovate UK, which is the UK government's innovation agency. And we got a good amount of funding through this precision medicine competition in 2018, and some of you are probably thinking, wow, that's 5 years ago, and it was 5 years ago, and I'll show you why it's taken 5 years to get from funding to where we are now in a minute. And our aim was to establish the first data commons for NAFLD research, and I'll tell you what a data commons is in a minute. And I was, for my sins, the academic lead for this project, and working very closely with a liver histopathologist called Tim Kendall, who has really been doing this with me over the last few years. And there was the consortium really consisted of Precision Medicine Scotland, which is a university-based precision medicine organisation in Scotland that helps people like me who want to investigate precision medicine projects to curate and store our data, particularly in a safe environment, and work in a safe, trusted research environment. The University of Edinburgh, Eagle Genomics, who are a bioinformatics company in Cambridge, they were part of this consortium, and also the network of biorepositories in Scotland, because as you'll see in a minute, we used and relied on liver tissue to start this whole program. So what is a data commons? Well, a data commons is basically a fancy database. It's a platform to integrate data from lots of different sources. We all know that data tends to sit in what we call silos in different databases, different places. And the idea of things like data commons is to try and put everything into one place so you can work with it and analyze it more effectively. So a data commons, for instance, could incorporate digital pathology images, fiber scans, any sort of omics data you want to layer into it, and data from electronic health records. And by connecting different digital objects and making them accessible, data commons can really catalyze research in different ways, and different ways that weren't possible before. So particularly for something like SteatoSITE, this hopefully will revolve around discovery and particularly validation. And I'm in Scotland, which is this chunk of the top of the United Kingdom. And the good thing about Scotland is that it has a single unified healthcare system. It has highly developed infrastructure for data science. It has a stable, pretty consistent population of around 5 million people. And this is quite good in terms of omics analysis on a national scale. I guess the key thing for health data science is that every single person in Scotland has a unique identifier called their Community Health Index number, or CHI number. And any contact they have with any part of the healthcare system is linked to this CHI number. So we have a way of connecting all the dots for each individual patient in terms of their healthcare interactions. And there is a long history of using electronic health records in Scotland. And this is facilitated by this network of what we call safe havens. So there are 4 safe havens in Scotland where all of the NHS data is basically funneled towards. So if you want to remove or extract any health data, you have to go to these safe havens where everything's done in a highly regulated way. And as a consequence, Scotland has some of the best health data in the world. And critically, it has a very high incidence of complex diseases for which NASH would certainly count as one. So what about Scotland and liver disease? Well, this is the European age standardized rate for chronic liver disease deaths. And you can see Scotland is much worse than the rest of the United Kingdom, and worse than most of other countries in Western Europe. Unfortunately, I haven't got Italy in this table, but I suspect you're somewhere in the mid-range here, but Scotland's far worse. And this is a graph showing obesity and overweight rates, and Scotland's for a small country is really punching above itself getting into the bronze medal position here in global obesity rates behind USA and Kuwait. And, what are the reasons for this? Well, part of it is, people in Scotland do eat, I'm generalizing here, quite unhealthy diets. So one of the delicacies in Scotland is the deep fried Mars bars. So it's a chocolate bar which goes into deep fat to fry it. I mean, they fry everything in Scotland. They even fry pizzas. So that's an issue. And also there is a big problem with alcohol in Scotland, and particularly young people drinking. The other thing that's curious about Scotland is that there is significant geographical variation. So if you look across Scotland and take the average, there are average here, there are areas where there's a much higher mortality rate from liver disease than others. And this is another way of looking at it. This is something called the Scottish Index for Multiple Deprivation. And this shows the areas in red are the most deprived areas geographically, and through to the ones in blue that are least deprived. And this isn't a big urban area here, so there's variation. But the thing about Scotland, there's a lot of rural deprivation as well. So if you looked at the whole of Scotland, you'd see a similar picture in very rural areas as well. So I think there's a big issue here with deprivation, and we know that fatty liver disease is something that's strongly linked to social deprivation. And in Scotland, deaths from liver disease are around 7 times higher in the most deprived areas compared to the least deprived areas. And this is something that we want to do something about. We want to improve service provision and access to liver disease treatment across the whole of the UK, not just in Scotland. Right, so how was SteatoSITE constructed? So we thought the best way of starting was to actually find tissue that we had a diagnosis for, had been curated and looked after and archived. So this was a tissue first case selection. So we went to the network of biorepositories in Scotland and tried to pull out as many cases with a histological diagnosis of non-alcoholic fatty liver disease, or NASH, as we could. And we went to the original biopsy referral documents. We went to all of the original biopsy reports. And obviously anyone that had any indication of alcohol anywhere was immediately excluded. And this here is this is actually on the floor of my colleague's office. These are all the glass slides from the SteatoSITE cohort. So there's quite a lot. And all of the blocks, the paraffin blocks from these tissues, were all centralized here in Edinburgh, and everything was recut and restained for H&E and picrocerius red in a single laboratory. And then every single slide was scanned, and each digital image we have curated, and all of these glass slides are available for rescanning. And we're about to rescan everything again because we have a new scanner. So we have the original glass slides, and every H&E and every PSR stained slide has been scored. So we had 3 consultant pathologists who all scored these for NAFLD activity score, and also the SAF scoring system for steatosis activity and fibrosis, the NASH CRN scoring system, and also a modified Ishak score for fibrosis. And we provided all these scores as part of the resource, but people may want to do their own scoring based on the images. We've also done digital quantification, looked at the collagen proportionate area and the fat proportionate area using open source software. We took thick curls of each block as well and sent that for bulk RNA sequencing. And that in itself was quite a challenge, because some of these samples are quite old, archival, paraffin, formalin fixed, paraffin embedded blocks of tissue. Having said that, out of 940 cases we managed to get hold of, 707 had valid RNA-seq, which I think is pretty remarkable. It's much more than I thought we'd get. And this includes 538 needle biopsies, 39 explants, and 130 liver resections. And this is after we've done some quite strict QC criteria to get rid of cases where we don't think that the RNA integrity or quality is suitable for RNA-seq. We also got around 40, well, we had 40 normal liver controls. These had been, again, scored histologically, and the absence of any fat, inflammation, or fibrosis confirmed histopathologically. And of those 40 normal livers, 34 are available for comparative RNA-seq analysis. We did also do RNA-seq variant calling to look for some of the common SNPs. As any of you will know who've done this, who've done variant calling from RNA-seq data, it is quite tricky. There's variable coverage, I would say, of some of these risk alleles, but nevertheless, the data is there, and other people may wish to repeat the analysis as well. Then the electronic health record data. I mentioned these safe havens earlier on. So there's one in Glasgow, there's one in Edinburgh, there's one in Aberdeen, and there's one in Dundee. So we have this network of safe havens, and what we wanted to do was to get time-stamped routine clinical data retrospectively from the electronic health record. So we took the date of the tissue sample, whether that be a biopsy or an explant or a resection, as our anchor point or time zero, and then we collected a variety of data before and after the tissue sample date, and we collected some static data, demographic data, age, sex, ethnicity, etc., deprivation index, and then we collected hospital coding data for comorbidities, for inpatient, outpatient episodes, routine laboratory data, fibroscans where they had occurred, any intervention. So we had all the prescribing data going back before and after the tissue sample date, and then cancer registry data and death data. And so for each patient, what we created is a data annotated timeline. So for instance here, so this is the time zero anchor point, and we've gone back in time up to 10 years before and collected data, and we've gone from the year 2000 to 2019 collecting data forward. So this is all retrospective, but you can create, if you like, pseudo longitudinal follow-up and these individual patient timelines, each of which is completely different. And so we have cases from 2000 to 2019 and data from 10 years before up until 2019. So in total, there's 5.67 million days of comprehensive clinical data that we have collected. Overall, the median follow-up post-tissue acquisition is around 7 years. So it's quite good amount of follow-up data. But what do you do with the data? This is one of my favorite scientists. His name's Ben Goldacre. He's the director of the Bennett Institute for Applied Data Sciences in Oxford. And he came up with this brilliant quote, which I think it just says it all, really. So this was talking about NHS Digital, which is the way of using digital health records in the UK. So what he said was 73 years of complete NHS patient records contain all the noise from millions of lifetimes. Perfect, subtle signals can be coaxed from this data, and those signals go far beyond mere academic curiosity. They represent deeply buried treasure that can help prevent suffering and death around the planet on a biblical scale. It is our collective duty to make this work. Now, he's used hyperbole, but I think he absolutely hits the nail on the head, which is there's so much data there that we can use, but how do we get hold of the data? How do we work with it? How do we pull out the important signals? So this is our attempt at trying to collect, curate, and analyze a very large data set. So this is if you like, an overview of SteatoSITE. So again, I won't go into this in too much detail, but suffice to say, here we start with the biorepositories and we identify the cases. We then do the pathology analysis, we do the transcriptomic analysis, and we do the electronic health record data analysis, so using coding data and other databases and repositories. Everything is then incorporated into a single place, a single server at Precision Medicine Scotland, the SteatoSITE database. And we have all of these different items of data, or everything related to time zero, or the time of tissue sampling. And we needed a huge amount of approvals, as you can imagine, and ethical approvals and things to get hold of this data. This is retrospective, unconsented data. So you have to be super, super careful with collecting and curating and storing this data, and for that matter, sharing it in a very careful way. And so we now have the ability to work with people who can explore the data as well. So in total, we were given 38 CSV files containing 1,450 columns and over 1 million rows, which needed processing. So we had a team of data IT analysts who have cleaned, restructured, standardized the whole data. That in itself took 1.5 years, I would say, because we had to go back to the safe haven, say this is wrong format, this needs tidying up, and this is headed wrongly, this is coded differently. So that was a huge task in itself. And we've ended up at the end of that with 534 distinct columns of data. And this is some very kind of high level view of the cohort. So the average age is around 55, pretty equal sex distribution. The one thing I would say is this is very much a white Scottish population, so it's probably not ever going to be the best cohort to make generalizations to different geographical areas or ethical groups, but nevertheless we can learn a lot from it. The mean BMI was 31, so most patients were obese or overweight, as you would expect. About a third had diabetes. And then just going back to this deprivation index, I think is important. It gives us an opportunity to look at the data, explore the data, both disaggregated for sex, but also disaggregated for other things such as deprivation, to look at how this affects outcome. So we have 940 cases covering the whole disease spectrum, and the majority of them are needle biopsies. Others are resections and explants. And so all the histopathological features have now been scored, and this is something called an alluvial diagram, which I just like the colors really, but what it shows is the relationship between the different histopathological features, so steatosis, inflammation, ballooning, and fibrosis, how they relate to each other. This is probably more interesting for you. This is the correlation between the continuous variables of fat percentage and picrocereus red collagen percentage with ordinal scoring, and this is kind of what you would expect. It's a sense check, if you like, that what we're dealing with is valid data. And then the really interesting thing that we did straight away was to look at outcomes. So basically, did our graphs look a bit like the Sanyal graphs for the prospective NASH cohort? And I guess in a nutshell, yes, they did. So people with more severe fibrosis had a much higher chance of death than people with milder fibrosis, and we actually had quite a large number of deaths in this cohort, but definitively here showed that scarring predicts all-cause mortality. And if you use an unbiased clustering algorithm, actually the F3s and the F4s cluster together, which I think is interesting from a sort of biological point of view, because the point where you get bridging, fibrous bridging, we think is a kind of inflection point histologically in the natural history, and that seems to play out in terms of outcomes, risk of outcomes as well. So what about decompensation events? Now, this is very important, I think. We again had quite a lot of decompensation events in this cohort. We used a composite outcome, and we used all the ICD-10 codes that were listed in this really helpful paper from Hannes Hagstrom and colleagues, where they really gave us a consensus statement on how to use administrative data for NAFLD research. So which codes do you look for to make sure you capture all the relevant outcomes? So we did that. We also added in procedure codes for things like banding of varices or paracentesis, etc., that clearly link to decompensation. And so our aggregated outcome of decompensation events identified 291 cases out of the ones who had biopsies. We only looked at the biopsies for outcomes because obviously people who've had an explant for end-stage liver disease are a very, very different group, as are the resection group as well. So this is just the needle biopsies we looked at here. And this is, again, this is the Kaplan-Meier graph, similar to the last one, showing that, not surprisingly, much higher risk of decompensation events in patients with cirrhosis. And then we had a relatively low number of HCCs, but that's understandable. It's an uncommon clinical outcome, but nevertheless, we get a similar type of graph, just more staggered because of the lower numbers. Then we've begun to look at the transcriptomics, and here this is a principal component analysis plot, just showing here really that you get clustering according to fibrosis stage. And again, you know, we've developed a number of heat maps here. This is looking at comparing the top 50 genes from F4 compared to F0 and F1, and you can start to look again across the different histopathological variables. And then we've done some pathway analysis, showing, not surprisingly, these are the top upregulated pathways. You'd expect to see scarring signals here, but I guess the thing that's interesting also, given that many of these patients get cardiovascular disease, we see lots of pathways for cardiomyopathies coming up as well, particularly as the fibrosis stage increases. So this dot plot here, the size of the plot corresponds to the gene count, and the color to the pathway enrichment significance. And this is something called a chord diagram, which looks pretty really, but this looks at the topmost differentially expressed genes for the GO terms, the gene ontology terms concerning collagen fibrils, extracellular matrix, wound healing, and vascular wound healing. And then something that we've developed, which, you know, hopefully the whole community will be able to use in due course, is we've created a web-based gene browser using an R-Shiny app. So you can basically type in any gene that you're interested in, and you can generate plots and things just to get a feel for what the database contains, what it looks like. Is your favorite target there? I've just put in collagen 1 here, and you can generate heat maps and other things. And this is a way of people just to get a handle on what is or isn't in the database, and you know, might encourage people to come and want to collaborate and so forth. So this will eventually be housed on the SteatoSITE website, and in our paper that we hope to get published relatively soon, there'll be a link as well to that. So what are the opportunities for further study here? Well, obviously we want to use this cohort to establish robust associations between the histology, the RNA-seq, and clinical outcomes across the whole NAFLD spectrum. I think it has got significant potential utility as a validation data set. So other people may want to say, well, you know, we've got this cohort, we found X, Y, or Z, we want to look in yours. We've started to use more advanced bioinformatics and artificial intelligence, machine learning approaches to try and, you know, we've got a lot of data here. How can we start to navigate that and pull interesting things out? I think it's a good resource for particularly looking at prognostic biomarkers. We'll be presenting some data at EASL, where we've looked at transcriptomic signatures and prediction of outcomes. You can start to use the data to develop clinical decision support tools, and this is going to be very important to refine care pathways so we can focus our efforts on patients at a more higher risk. Really important role for this potentially is discovering and validating drug targets and combinations, and also training new digital pathology algorithms. We've got this whole 940 case library, if you like, of curated digital images, so people can apply new digital pathology algorithms, and that's something again that we will hopefully be presenting at EASL. So what have we done so far? Well, one thing we have looked at in collaboration with others is predicting the risk of HCC and NAFLD. As I mentioned before, it's a low incidence outcome, and it can occur in non-cirrhotic livers as well, which is slightly curious. And it's commoner as the number of metabolic conditions increase in an individual. And so we were approached by an Australian group who had done some lovely single-cell RNA sequencing work, and they'd identified a hepatocyte phenotype that predicts HCC potential. And they'd done mouse models, and they were looking to validate their signature in human cohorts. So they basically used their single-cell RNA-seq database, and they used bulk RNA-seq from our data set and other data sets, and did deconvolution analysis to estimate the number of these disease-associated hepatocytes, or DARHEPs, in the livers. And so we were very happy to use our database with them, and they again looked at the frequency, estimated frequency of these disease-associated hepatocytes across the fibrosis stages in SteatoSITE, and then generated a ROC curve, and then generated UMAPs of high and low DARHAP frequencies. And then they used the RNA-seq data again to look at fibrosis-related genes, ductular reaction-related genes, and inflammation-related genes. So it's a nice example of how we can collaborate with other people to validate observations. We got another grant from Innovate UK a couple of years ago to use AI to try and interrogate the data. So we work with this company called Bering, who are based in England. Bering have this proprietary AI platform called Brave AI, and we've been working with Bering to look at the data and allow us to predict outcomes. So they created a model where they had a 10-year observation window within the database, and then a prediction window within the database. And we started looking at all-cause mortality. And although they had access to everything, including some static data points, but also other longitudinally collected data points, you'll see from the data that although you get really good numbers with everything, when they just used a limited number of coding outcomes and some static demographic data, you also get very good statistical performance. So what they showed here is using everything, basically, you can get very good prediction of 3-month all-cause mortality. As I said, you still get OROCs of around 0.8 and above using a much more limited data set. So it begins to show you what you can do when you start exploring a big data set with AI. And we're now validating this original deep learning model that we used in an independent NAFLD cohort. Then another thing that we've been interested in doing, and we got another small grant for this, was to try and identify tissue biomarkers. So we have lots of tissue, and we have a way of analyzing the tissue. So we use single cell data reference set, and we use SteatoSITE, and we use this multi-subject single-cell deconvolution. It's running away with itself. Program called Music to estimate the cell proportions in the SteatoSITE tissues. And I've just focused here on mononuclear phagocytes because that's a cell type we're interested in, and particularly scar-associated macrophages, which we know are enriched in areas of scarring and fibrosis. And here we've used cell type proportions to predict outcomes. And here you can see, sorry, it's done it again. You can see here an increased risk of decompensation as the proportion of, according to the proportion of scar-associated macrophages. And then if you identify a pathogenic cell type, you can then look for cell markers to identify those cells in tissue. And this is a multiplex panel we've been developing to look for these scar-associated macrophages. So another way of being more refined in our histological analysis. So digital pathology, again, is an area of intense interest in the field. And we've been working with HistoIndex using their second harmonic generation 2-photon analysis, and they published this data authored by Nikolai Naumov a year or so ago, showing that digital pathology spots things that conventional pathology misses. And so I think this is in itself is very interesting, but then the next thing to do is to try and link some of these novel histological metrics, digital histological metrics, and try and link these to outcome. And I think that's the next big thing. And certainly we're going to present some data at EASL, again, working with HistoIndex, looking at their digital pathology metrics and how that might predict outcomes. Then another obvious thing to do is to use SteatoSITE to identify novel treatments. So I have a PhD student called Maria, who's funded by MRC, and she's been looking at using SteatoSITE and using Connectivity Map and other drug databases, and using computational techniques to try and identify drugs that may correct the abnormal transcriptional signatures that we identify across the disease spectrum. She's also tried to develop an in vitro model of NASH using iPS cells. So she's developed these trisphere model in 3D, consisting of iPSC derived hepatocytes, stellate cells, and endothelial cells. And this hopefully will create the environment to test some of these drugs and drug combinations that she identifies computationally. And the interesting thing that we've done is we've looked using nanostring at the sort of RNA expression analysis in the spheres, and compared that to SteatoSITE database as a reference. And what you'll notice, even those of you that aren't that used to looking at heat maps, is that there's more similarity between the spheres, in vitro spheres, as the F4 patients, and some of these genes that we picked out that are highly expressed in both, like the aldo-keto reductase family 1 member B10, for instance, which is quite well validated in NASH, gives us some confidence that these models are probably telling us things that are relevant to the human in vivo situation. So basically, that's what we have. It's been a lot of work. I hope we're now at the point where we can start to collaborate with people. We can start publishing things, presenting things. And I think there's lots of different ways that the resource can be useful. And if I haven't covered anything so far, you can ask me questions, or if anyone's at EASL, I'm happy to speak then. And these are all the people and funders and things that I think I need to recognize. So I'm going to stop sharing my screen now and give you guys some time to ask any questions that you might have. And I apologize again for not presenting everything I can present, like some of the risk prediction stuff, some of the digital pathology stuff, but I'm embargoed by EASL, unfortunately.
Well, thank you very much, Jonathan. Fantastic job.